Ascomycota is a phylum of fungi that undergoes both a sexual and an asexual life cycle. When we talk about sexual life cycles, we refer to them as teleomorphic, but when we talk about asexual life cycles, we refer to that as anamorphic. So anamorphic is asexual and teleomorphic is sexual. Right here, we're going to talk about the teleomorphic or the sexual life cycle of the Ascomycota, also known as the sac fungi. And we will also learn the reason behind the name of this phylum of fungi. So, just like any other fungi, we start off with a spore. And in this case, we start off with a spore that is N. So remember that N refers to the ploidy. So whenever we see N, that is going to be haploid. But when we see 2N, that's going to be diploid. So we start off with a haploid spore, and we notice that it actually has 2 nuclei. Okay, so it has 2 nuclei, and they are the same nuclei. So in a way, we could say that they're sister nuclei. Now this spore is referred to as an ascospore. So if we spell that out, it's ASCO spore. Now this haploid ascospore with 2 nuclei, it begins to germinate. And when it begins to germinate, essentially what happens is that notice we see that the nuclei start to push towards the edges and produce these hyphal structures. So as germination continues, we can see that we get these compartments. We get this septation happening between each nuclei, and as a result, we have this haploid mycelium. Now, what's interesting about the sexual life cycle of an ascomycotan fungi is that we have both a female part and we have a male part. Now, the female part, which we're going to denote in green over here, is known as the ascogonium, and the male part is actually known as the antheridium. Now, what happens is that, first of all, we can notice that this is still N, it's still haploid, but all of a sudden, we get this swelling. And when this swelling occurs, we see that all the nuclei in this compartment are all together with no septation. So remember, the septation that we see over here, it's no longer present in this tip swelling. Now that swelling is what we refer to as our ascogonium on the female. And over here, this is the antheridium swelling. Now, the female part will extend out this tube-like structure to the antheridium. This tube-like structure, represented in blue over here, is called the trichogyne. The trichogyne, if we break it down into its root words, tricho means hair, and the gyne part means female. So essentially, we're saying female hair. But it is essentially referring to this tube that extends from the ascogonium into the antheridium, and it pulls the nuclei from the antheridium into the ascogonium. So the nuclei, we can see this arrow, the nuclei are moving in this direction. But remember that the trichogyne, the tube, it was produced by the ascogonium to the antheridium. And then the nuclei move from the antheridium to the ascogonium. And as a result, we get this mixing of nuclei in the swollen compartment. And now we have N plus N. We are no longer haploid because now we have 2 sets of nuclei that are different. So we have the orange nuclei and we have the green nuclei. So these are 2 different nuclei. That is why it is N plus N, and we are no longer simply N. This N plus N stage is referred to as dikaryotic. When we only had that single nuclei, that haploid nuclei, although we had 2 nuclei, but they were sister nuclei, they were the same, they were haploid, we referred to this as monokaryotic. We have we have 1 type of nuclei. But now we have 2 different types of nuclei, N plus N. We are dikaryotic. We have 2 nuclei. Di means 2. So in this swollen compartment, we get this mixing of nuclei. But remember that in the other compartments, we're still going to be haploid. We're just seeing this mixing occur in that swollen compartment. Now, over here, we have the green nuclei down here, because remember that the nuclei moved from the antheridium to the ascogonium. So if it moved from here to here, whatever was down here is going to remain the same, right? So that's why we still see the green nuclei over here and not the orange one. Now the next step is that these nuclei, they are going to push towards on the swollen compartment to produce these Hyphal structures. And these Hyphal structures are going to be known as the ascogenous hyphae. Now, what does ascogenous mean? We can break down this term once again. Asco simply means sac, and genus, well, genus is generate or to create. So we are going to create these sac-like hyphae, and we'll get into that in a minute. But basically, we get the nuclei pairing, so we can see green and orange, and they're pushing towards this edge over here. And we get these Hyphal structures forming, the ascogenous hyphae. Now, if we zoom in to any one of these Hyphal structures, for example, let's zoom into this one. It doesn't matter because they're all the same. And we see that it is n plus n. Now, the question now is, how do we extend this Hyphal structure in order to continue making compartments, in order to continue Hyphal growth? Remember, we need to produce more nuclei. So essentially, we're going to undergo mitosis. We're going to undergo mitosis, so we're going to get 2 green nuclei and we're going to get 2 orange nuclei. But remember that whenever we see mitosis within fungi, it is very common that septation, because remember, these compartments, they're separated by this septation, right? So how do we get that septation? Well, commonly in fungi, septation occurs at the metaphase plate. Now, let's try to think about why it happens at the metaphase plate. Well, we know that whenever mitosis or meiosis is occurring, we have those actin filaments, we have microfibrils, microtubules, we have all of these different types of structures that are utilized in pulling the nuclei apart. But at the end of metaphase, we have some of those structures left. We have some actin filaments left, we have microtubules left, and those remnant structures are what are used to create that septation between the compartments of these nuclei. So when mitosis occurs, so let's say mitosis, when it occurred right over here and we got these 2 sister green nuclei, at the metaphase plate, we have remnants of those microfibrils, microtubules, etc. And at that metaphase plate, we will see septation occur. Same for the orange ones. We see this septation occur here too. But now we have a problem. We need our hyphae to be n plus n. But if we take a look over here, we have n plus n. We have green and orange. But over here, we only have green, and over here, we only have orange. How do we fix this problem? We need to be n plus n. Well, first of all, we have to name this structure that I forgot to mention earlier. But when this mitosis occurs, so when we get the duplication of the nuclei, at the same time as meiosis, we get this curving of the hypha. This curved structure over here is referred to as the Crozier. I'll spell this out. So it's Crozier. And that's this bending structure over there. Now, this bending structure, it continues to bend, and it joins up with down over here. As it joins up, the cell wall that was on the outside over here dissolves, and that nuclei, the green nuclei, is going to be released into this compartment. So essentially, that green nuclei is going to move into here. So what was the benefit of having this Crozier? it's essentially very similar to a clamp connection. But instead of having that clamp connection come out from one of the compartments and reach over to the other compartment, we have at the tip the Crozier, which bends at the same time as mitosis is occurring, and it drops in that nuclei into that compartment that only had the single nuclei, and now we have N plus N in all of our compartments. So what's the next step? Well, the next step is going to be karyogamy. So karyogamy is going to be when two nuclei are going to come together. So we can see, and it's going to happen in the tip. So in the tip, we can see we have N plus N, we have a green and orange nuclei, and they're going to come together to form this zygote, which is going to be 2N. So we can see it's 50% green, 50% orange, and we have this 2N structure. So immediately after karyogamy, we are going to undergo meiosis. So we're going to undergo meiosis 1 and meiosis 2 to get 4 haploid nuclei. Okay, so now we have 4 haploid nuclei, and the next step is that the nuclei, a cell wall begins to form around these nuclei. So over here, we'd actually see cell wall formation around each of these nuclei. But we're not done yet. If we take a look over here, our ascospores are haploid, but they have 2 sets of nuclei that are the same. Over here, we only have 1 set of nuclei. We're still haploid, but we need 2 sets of nuclei in order for these spores to be released. So what happens is that the nuclei are now going to undergo mitosis to get the 2 nuclei. Now, this mitosis step is referred to as post-meiotic mitotic division. So remember, post means after. We just underwent meiosis, and now we're doing mitosis. So post-meiotic mitotic division is represented right over here. And then once this happens, this structure over here begins to dissolve, and these ascospores are now released, and the cycle will continue. And that is essentially the life cycle of the sexual life cycle of the Ascomycota.